Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing Odisha CPET Biotechnology question paper conducted in the year 2020. So coming to the first question. Peroxisome is involved in. So it is involved in detoxification from free radicals. Answer is option B. Then next one. Which is wrong about plasma membrane? It divides the internal space into discrete compartments. Yes, that is correct. It organizes complex reaction sequences and conserves biological energy. That is also correct. It allows free movement of all nutrients for growth and division. This free movement, it allows free movement. It doesn't allow free movement. So this is the right answer. Option C is the right answer. It has a central role in cell-to-cell communication. That is also correct. Then which is not a part of prokaryotic ribosome? It is 18S rRNA that is not a part of prokaryotic ribosome. Which organelle is not associated with photorespiration? All these are direct questions, okay? Mitochondria, peroxisome and chloroplast, those three are involved in photorespiration, whereas lysosome is not involved in photorespiration. Then triggering of the intrinsic pathway of apoptosis involves a balance between pro-apoptotic and anti-apoptotic proteins. Which of the following is anti-apoptotic? So the intrinsic pathway of apoptosis, it involves two pathways, that is not two pathways, a balance between pro-apoptotic and anti-apoptotic proteins. So which of the following is anti-apoptotic protein? So out of this, BCL2 is anti-apoptotic protein. Then enzyme immobilization is imprisonment of enzyme to a support or matrix. So what does it do? It provides enzyme with a protective covering. So answer is option B. Then genetic engineering is possible because we can cut DNA at specific sites by endonuclease like DNase 1. No. Restriction endonucleases purified by bacteria can be used in vitro. Yes. That is the reason why genetic engineering is possible. Then next one. A plasmid cloning vector should contain all of the following sequences except. So origin of replication, yes, that has to be there. Selectable marker gene, yes, you want to select after you have done your recombination. Multiple cloning site also should be present. But inducible promoter, that is not necessary for plasmid cloning vector. Inducible promoter, it is the characteristic feature of operon where its function is to promote protein synthesis. So answer here is option C. Then which of the following DNA sequences is recognized and cleaved by a restriction enzyme? Now the restriction enzyme, it will cleave palindromic sequences, that is nucleic acid sequences where the one strand matches its complementary strand when read in the same direction. That is, if one strand reads from 5' to 3' direction, the same, it should be its complementary strand should read the same in 5' to 3' direction. So here if you see B, that is GATATC, and this is the complementary strand CTATAG. Now if you read from here, that is okay, from this direction, that is 5' 3' direction, the complementary strand should read the same in 5' 3' direction. That is, in this direction it should read the same. So here GA, so here also GA, TA, TA, TC, TC. So answer B is the correct answer. Then BT cotton is widely cultivated. BT cotton means it contains endotoxin gene of BT. Bigger thread variety, no. Cotton seeds treated with barium, no. Produced by biotechnological enzymes, restriction endonucleases, no. Contain endotoxin gene of BT, yes, that is correct. BT cotton plant contain one or more foreign genes derived from the soil bacterium Bacillus thuringiensis. And insertion of the genes from Bacillus thuringiensis causes the cotton plant to produce what is called as crystal insecticidal proteins or cry proteins. Then next one. Transgenic plant flavor saver tomato carries an artificial gene for delayed ripening process, yes. Longer shelf life, yes. Added flavors, yes. All of these. So flavor saver tomato, it is genetically modified tomato and it was developed by antisense RNA technology. And it was modified to regulate the expression of the enzyme polygalacturonase. Then next one, genome is the entire set of DNA instructions found in a cell. So it includes your nuclear DNA, mitochondrial DNA as well as chloroplast DNA. So the answer here is option D is the correct answer. Now reverse transcriptase. So reverse transcriptase is an enzyme used to generate complementary DNA from an RNA template. So RNA is the template strand. So it should be RNA dependent and DNA is produced. So DNA polymerase. So the answer here is RNA dependent DNA polymerase. Answer is option B. The next one. If a hybrid DNA molecule labeled with N15. So I have labeled the other strand with N14. That is a hybrid DNA molecule. Is allowed to replicate twice in normal culture medium. So this is first round of replication and this is second round of replication. The percentage of hybrid DNA after second replication. So what will be the percentage of hybrid DNA after second round of replication? So DNA replication, it is semi-conservative. So one half will give rise to N14 and the other strand will give rise to N14-15 because it is allowed to replicate in normal culture medium. So the culture medium is having N14 DNA, not N15. Now it undergoes the second round of replication, that is also semi-conservative, and this is the percentage of hybrid DNA you have to calculate. So if you take 100%, the hybrid DNA is only 25%. So answer is option C. Then next one. Melting temperature value of DNA is high when it contains. So GC pair has 3 hydrogen bonds while the AT pair has only 2. Okay, before that I will tell you the answer. It is G plus C greater than A plus T because GC pair has 3 hydrogen bonds and AT pair has only 2 hydrogen bonds. Therefore, GC pairing is more stable than AT pair and the strands with more GC content have more hydrogen bonding, are more stable and have greater resistance to denaturation. Therefore, they require higher melting temperature to decompose. So G plus C is greater than A plus T. Then proteomics is the study of. Now proteomics is the large scale study of proteomes. Now what is a proteome? It is a set of proteins produced in an organic system or produced in an organism. So option A, if you see entire set of expressed proteins in a cell, yes. Set of proteins, yes. Set of proteins in a specific region of the cell, yes, that is also a proteome. So answer here is option D, that is all of these. Then the genetic code is called degenerate because. Now degenerate means a single amino acid may be coded by more than one codon. So one codon has many meaning. No. One codon doesn't code for single amino acid alone. One codon doesn't code for so many amino acids. It is one amino acid that is being coded by more than one codon. So more than one codon has the same meaning. Yes. So option B is Correct. Now, possible blood group in children from the parents with BO blood group are. So this is capital B, capital O. I have not put IB, IB, IO, IO. In order to avoid confusion, I have wrote it as BB and O. Now, if the parents are capital B, capital B, capital O, you will get two gametes only from each one gamete from each of the parent, and you will get only one offspring or all the offsprings with BO. Now, it can also happen that the B blood group has got the genotype BO, whereas O blood group, it is always recessive genotype, so it will be OO. So here you have got B and O as the gametes, and for O blood group only one single gamete is produced, and you will get BO and OO. So what are the possible blood group in children with parents with B and O blood group will be both B and O. Answer is option C. Gynandromorph or sex mosaic is produced by. So gynandromorphs are individuals who contain both X0 and female XX characteristics. The male character is present in one part and the female character in another part. In Drosophila, it results due to the loss of one X chromosome during the first meiotic division. So the answer here is option A, that is loss of X chromosome. Then next one, embryonic stem cells are derived from. So embryonic stem cells are derived from the inner cell mass of the developing blastocyst stage embryo 5 to 8 days after fertilization. So it is undifferentiated. So answer is option A, that is undifferentiated inner cell mass of embryo. Next one, after which cleavage blastocyst is formed, how many cells are found in it? So after fifth cleavage, blastocyst is formed and around 32 cells are found. Answer is option C. Then synovial fluid contains. So synovial fluid, it is produced by the synovium and is composed of water, inorganic salts, macromolecules, hyaluronic acid, lubrine and aggregants which contribute to lubrication. So the answer here is hyaluronic acid. Only one component they have given. Option B. The next one. Which of the following hormones is or are helpful to regulate blood pressure? So if you see for ANH, ANH is atrial natriuretic hormone, is to lower blood pressure and to control electrolyte homeostasis. Another is aldosterone. It is responsible for regulating sodium homeostasis, thereby helping to control blood volume and blood pressure. So the answer here is option D, that is aldosterone and ANH. Then next one, tricuspid valve is found in between. It is found in between right auricle and right ventricle. Option B is the correct answer. Then antigen antibody reactions are. So it can be, they are reversible. Antigen antibody reactions are reversible as well as they are specific. So as per me, both A and C are the right answer. Then next one, the reaction of soluble antigen with antibody is known as. So remember, it is soluble antigen with antibody. Okay, now what is, the answer is precipitation. Now what is agglutination? Agglutination is antigen antibody reaction in which particulate antigen combines with antibody to form visible clumping of particles. Whereas in precipitation, soluble antigen combines with soluble antibodies and to form insoluble visible complex. This is called as precipitation. So answer here is option A, precipitation. Then monoclonal antibodies are produced by, direct question, very easy, hybridoma technology. Answer is option A. Then antibodies in our body are produced by B lymphocytes. T lymphocytes produce T cells. Sorry, okay. Then the cellular immune response is mediated by, so cellular immune response is mediated by T cells and humoral response is mediated by B lymphocytes. So the answer here is option B, that is T cell. Then tautomerization, it is the shift of hydrogen, not shift of carbon, it is shift of hydrogen. So answer is option A. Then which one of the following is a rate limiting enzyme of gluconeogenesis? So here is the pathway of gluconeogenesis. It is formation of glucose. Now this arrow, downwards arrow, they are for glycolysis. And reverse of glycolysis is gluconeogenesis. So in gluconeogenesis, 3-phosphoglycerate is converted to 1,3-bis-phosphoglycerate by phosphoglycerate kinase. Then pyruvate is not directly converted to phosphoenol pyruvate. You need pyruvate carboxylase to convert to OAA and phosphoenol pyruvate carboxylase to convert to phosphoenol pyruvate. Then the other fructose 1,6-biphosphatase, even though it is a reversible reaction, it is catalyzed. Both the forward and reverse reaction are catalyzed by different enzymes. So fructose 1,6-biphosphate is converted to fructose 6-phosphate by fructose 1,6-biphosphatase, and fructose 6-phosphate by glucose and glucose 6-phosphate is converted to glucose by glucose 6-phosphatase. So which of the following is a rate limiting enzyme of gluconeogenesis? Is it phosphofructokinase? No. It is involved in glycolysis. Hexokinase also involved in glycolysis. Pyruvate kinase is also involved in glycolysis, whereas pyruvate carboxylase, it is involved in gluconeogenesis. So answer is option C. Electron transport and phosphorylation can be uncoupled by compounds that increase the permeability of the inner mitochondrial membrane too. Okay. Compounds that increase the permeability of the inner membrane to H positive allow electron transport to proceed but inhibits ATP synthesis. Hence, electron transport and phosphorylation, they can be uncoupled by uncouplers such as 2,4-dinitrophenol. So, electron transport and phosphorylation can be uncoupled by compounds that increase the permeability of the inner mitochondrial membrane to protons. That is what uncouplers do. Answer is option B, that is protons. The next question. Substrate level phosphorylation in TCA cycle is catalyzed by. So succinyl-CoA synthetase is the only mitochondrial enzyme capable of ATP production via the substrate level phosphorylation. And succinyl-CoA synthetase is also known as succinate thiokinase. So the answer here is option D. Then next one. Synthesis of glucose from amino acids is termed as. So when glycogen stores are depleted and glucose is required by the body, the liver initiates the production of glucose from amino acids, lactate and glycerol in a process called as gluconeogenesis. Answer is option B. Then the correct sequence of cell organelles during photorespiration is, so for photorespiration you need chloroplast, mitochondria and peroxisome. Now the correct sequence is chloroplast, peroxisome and mitochondria. Answer is option A. When the leaf of the touch me not plant is touched, the leaf drops because. So when leaves of Mimosa pudica, that is touch me not, is touched, the stimulus reaches the base of the leaf. Owing to this stimulus, the turgor of the lower half of the pulvinus is lost and the leaf droops down. So the answer here is water is lost from the cells at the base of the leaf. Then velamen cells are found in? So velamen, it is a spongy multiple epidermis that covers the roots of epiphytic plants. With the help of this velamen, they are able to absorb water from the dew, moist atmosphere and also rain. So answer here is option A, that is epiphytes. The condition of polyadelphous is found in family. So polyadelphous condition, the stamens of the flowers are fused by their filaments while the anthers are free. So it is found in citrus family, that is Rutaceae. Answer is option A. Then which of the following is not a function of auxin? Inducing callus formation, yes. Inducing dormancy, yes. That is not a function of auxin. Enhancing cell division, yes. Maintaining apical dominance also, yes. Then nitrite are oxidized to nitrates by a microorganism Nitrobacter. Answer is option C. The difference between gram-positive, gram-negative bacteria is shown to reside in the cell wall. Answer is option A. Which symbiotic bacteria is nitrogen fixative with the root nodules of leguminous plants? So with the root nodules of leguminous plants, it is Rhizobium. Answer is option D. Then microorganism used for alcoholic fermentation. It is Saccharomyces cerevisiae. Answer is option B. The bacterial cells are at their metabolic peak during. So metabolic peak is during log phase. Answer is B. When the power of ocular lens is 10x and objective lens is 20x, the magnification is, so total magnification is objective magnification into ocular magnification. So 10x into 20x, that is 200x, or it is 200 times. Answer is option C. Then which of the following is used to visualize live cells? So two most common methods for visualizing live cells is phase contrast microscopy and differential interference contrast microscopy. So all they have given is phase contrast microscopy in the option. So answer is option C. Then in gas chromatography, the basis for separation of components of volatile material is the difference in. So it is based on the difference in partition coefficients. Answer is option A. The basis of separation of the components of the volatile material is the difference in the partition coefficient of the components as they are carried through the column by an inert gas such as helium. So the answer here is option A, that is partition coefficient. Then ion exchange chromatography is based on the, so ion exchange chromatographic separation method is based on the net charge of the protein or based on electrostatic attraction. Answer is option A. Then if proteins are separated according to their mobility, then the type of electrophoresis is. So in SDS page, the proteins are separated according to their electrophoretic mobility. So answer here is option A, that is SDS page. The next one, T half or half-life of an irreversible first order reaction S to P is 1 day. Time required in days for the completion of 75%. So T half is 1 day. Decay constant is 0.693 divided by T half. So it is 0.693 per day. The time required for 75% conversion. It is given by T is equal to 2.303 by K log to the base 10 A minus AX. A by A minus X. So upon solving and substitution and solving, you will get the answer as option 2. So the answer here is option B, that is 2. Then next one, NMR is based on, so NMR is based on magnetic moment of the nucleus. Answer is option C. Then weakest force is, it is van der Waals force. Answer is option A. Covalent bonding between two molecules requires. So a covalent bond consists of the mutual sharing of one or more pair of electrons between two atoms. So for that it requires electrons with opposite spins. Answer is option A. Then radioactive substance emits the following rays except. So beta, gamma, alpha, all these are emitted by various radioactive substances. X-rays are not emitted. So answer is option D. Then which of the following is used as electrolyte in a dry cell? It is ammonium chloride and zinc chloride. Answer is option A. The next one. If the radius of the earth were to shrink by 1%, its mass remaining the same, the value of g, that is acceleration due to gravity on the earth's surface would be. So the acceleration due to gravity, it is given by g is equal to gm by r square, or g is inversely proportional to 1 by r square. So r decreases by 1%, so the new radius will be r minus 1 by 100 r, or it will be 0.99 r. Now the new g, that is g dash, it will be gm by r dash square. So substitute it and you will get g dash is equal to g into 1.020 or 1.020 g. Now it changes, it will be 1.020 minus 1, so 0.02, or it will increase by 2%. Answer is option B. Then electric current is measured by a meter. Answer is option D. Diffusion of light in the atmosphere take place due to dust particles. Answer is B. Then a body must be lifted to how much height so that the body gains an equal amount of potential energy and kinetic energy moving at a speed of 20 meter per second. So a body has to be lifted in such a way that it will have potential energy and kinetic energy equal. So here mgh is potential energy, half mv square is kinetic energy. So 9.8 h into half into, they have given the value of 20 v square. That is not v square, velocity it is 20 meter per second. So substitute that. So 20 into 20, you will get h as 20.4. Here the value is approximately near to the option B, that is 20.2 meters. Then two events A and B are mutually exclusive and each has a non-zero probability. If event A is known to occur, the probability of the occurrence of event B is 0. Answer is option C. Then correlation coefficient is a number between -1 and +1. The shape of normal distribution curve is bell-shaped. If hypothesis is rejected at 5% level of significance, it will always be rejected at 1% level of significance also. Then a list of 5 pulse rates. These are the values. What is the median for this list? So arrange the values in either ascending or descending order. See for the middle value, it is your median, that will be 74. Answer is option A. Then how many ways can 8 Indians, 4 Americans and 4 Australian persons be seated in a row so that all persons of the same nationality sit together? So 8 Indians can be arranged in 8 factorial ways, 4 Americans can be arranged in 4 factorial ways, and 4 Australians can be arranged in 4 factorial ways. So total number of possibilities as 3 sets is 3 factorial, and total number of ways in such a way that persons of the same nationality sit together is 3 factorial into 8 into 4 into 4 ways. So 3, 4, 8, 4. Answer is option A. Then next one, log A by B plus, it is not log B plus A, that's a printing mistake, it is B by A. Only then you will get log A plus B. Then A plus B is, so log A by B into B by A is equal to log A plus B. Log 1 is equal to log a plus b is equal to 1. Answer is option A. Then what is the value of x in the logarithmic equation? So I have solved it here. You can just go through it. The answer is 4. Then next one, in triangle ABC right angled at B, this is the triangle right angled at B. AB is 3 centimeter and BC is 4 centimeter. The value of cos C. So cos is side adjacent to angle C divided by hypotenuse. So before that we have to know the value of hypotenuse. So using Pythagoras equation we will get it as 5 centimeter. Now cos C is side adjacent to angle C, that is this one, this 4 and divided by hypotenuse 5. 5 is not seen over here in this photograph, but the answer is 4 by 5. I have written 5 but it is not visible over here. So answer is option D, that is 4 by 5. Then in an examination, a student scores 4 marks for every correct answer and loses 1 mark for every wrong answer. If he attempts in all 60 questions and secures 130 marks, the number of questions he attempts correctly are. So the number of correct answers be x. Then the number of incorrect answers will be 60 minus x. So 4x minus 1 into 60 minus x is equal to 130. Solve the equation. You will get answer B as 38. Option B, that is 38. Then next one. The expected value or dash of a random variable is the center of its distribution. Mean. The expected value or mean of a random variable is the center of its distribution. So it is mean. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Thank you.